Good morning, everybody. Okay, today we're going to talk more about karyotypes, specifically some types of karyotype abnormalities. But then we're going to go on and talk more about what we're seeing with a karyotype in terms of maps and genes. And we'll see some really interesting examples of some high-tech ways of looking for genes on chromosomes. Okay, so these are just some of the chromosome aberrations we're going to talk about during the semester, but these are the ones that you've probably heard about and that are the most familiar. Okay, so we have trisomy in a diploid means that we have 3 copies of a particular chromosome rather than the usual number, which is 2. Monosomy is a case where only one member of the pair is present, and therefore the person is short 1 chromosome. Okay, these are both examples of aneuploidies, where there's a deviation by 1 or a few number of chromosomes from the usual euploid number, as opposed to, for example, triploidy, where all of the copies are in extra form, as opposed to just 1. We will also talk about translocations, which involves rearrangements such that the chromosomes have exchanged parts with a non-homologous chromosome. Now, there are other types of chromosome abnormalities that also exist, but we're going to talk about those on a different day. Okay, so human karyotype trisomy. We can describe a human karyotype as having 2 N chromosomes. In the case of a trisomic cell or individual, we have its 2N plus 1 for a total of 47. For monosomy, the total is, of course, 45, and it's 2N minus 1. Okay, as I said a minute ago, euploid refers to complete sets of chromosomes, and those are haploid, diploid, triploid, etc. There's more than one way in which an abnormal karyotype can occur, the most familiar being the result of errors in meiosis, where a particular gamete gains or loses a chromosome relative to what they're supposed to have. Other events, though, are less familiar, and they occur during the lifetime of an individual. These are called somatic mutations. These result from errors in mitosis and or various types of DNA damage or chromosome breakage, etc. In those cases, the person is a somatic mosaic, meaning that some of their cells contain the abnormal karyotype, where other cells do not contain that. So important events can occur during your life, leading you to become a somatic genetic mutant in various ways. Okay, so here's a karyotype of a little boy who has Down syndrome, which is trisomy 21, and the extra copy of chromosome 21 is circled on the karyotype. And you can see it depicted in the ideogram. So this is a 47 chromosome person, 2N plus 1 chromosomes. Okay, monosomy X is the only example of a monosomy in human that's viable, and it leads to Turner syndrome. You can see on this karyotype, the woman has 1 X chromosome and no Y chromosome. So she's missing a chromosome, has only 45 total chromosomes. Okay, so in terms of aneuploidies, with the exception of trisomy 21 and the sex chromosome abnormalities, trisomies and monosomies in human are generally lethal before or shortly after birth. Turner syndrome, as I said, is the only non-lethal monosomy for reasons that we will talk about at some point later. There are some developmental problems with these females, but treatments are available. This is also described as the XO karyotype. Okay, euploid, we gave an example last time, is triploids, and they have a multiple of the normal number of chromosomes. So they have, to distinguish from trisomy, a triploid has 3 copies of each chromosome, not just 1 extra of one of the chromosomes. Okay, here's an example of a triploid that we saw last time, where there's 3 copies of each of the chromosomes with the same number of different chromosomes, which is to say 23 N in human. Okay, translocations may not be so familiar. Translocation means that a part of a chromosome has been moved to another non-homologous chromosome. They're often reciprocal, meaning that 2 different chromosomes have exchanged parts with each other. So it's a reciprocal in that both chromosomes have switched. Tumor cells are often aneuploid, and we'll talk about that later. CML, or chronic myelogenous leukemia, is the result of a translocation which has been known for a long time, which results in the formation of an abnormal chromosome called Philadelphia chromosome. It involves chromosomes 9 and 22 and a reciprocal exchange of parts between those two chromosomes. We'll talk about that in a sec. The other equally famous one involves Burkitt's lymphoma, which involves an exchange between chromosomes 7 and 14. These cause disease because the rejoined chromosomes create oncogenic forms of the ABL2 and BCL2 proto-oncogenes, respectively. So here's the CML translocation, a picture of it, and it can be detected, as we'll see in a little while. Now, what's happened is chromosome 9 and chromosome 22 have exchanged parts. So part of chromosome 22 is now found at the end of chromosome 9, and a little teeny bit of chromosome 9 is now attached to chromosome 22. So those two chromosomes have exchanged parts. Now, many times this kind of translocation is completely harmless, but in this particular case, it causes an unfortunate rearrangement where a proto-oncogene called ABL has been altered, so now it's constitutively active and made in great quantities, which creates an oncogenic form. Okay, so here's your CML karyotype as it's observed, where you can somebody can see a very shortened chromosome 22 and a lengthened chromosome 9, and it's the little 22 guy that's the Philadelphia chromosome. Okay, so what's happened, ABL is a proto-oncogene. The normal protein serves an important role in signaling, and its activity is generally low and very carefully controlled. As a result of the DNA rearrangement, an abnormal protein is formed that has excessive and uncontrolled activity, and this is oncogenic. And we can explain the molecular mechanism about that some other time. Okay, here's a few questions about karyotypes. A human baby's karyotype has 3 copies of chromosome 18, 2 X chromosomes, and no Y chromosome. Thus, the baby has blank total chromosomes and is blank. A, 46 female, B, 47 female, C, 46 male, D, 47 male. Okay, so the answer is B, right? Right. Okay, a baby has 3 copies of chromosome 18. His karyotype can thus be described as trisomic, monosomic, aneuploid, A and C, B and C. Okay, it's D, right? He's trisomic and also aneuploid. Which of the following is caused by a reciprocal translocation occurring in somatic cells? Turner syndrome, CML, trisomy 18, Down syndrome. Okay, so the answer is B, right? Right. Okay, so now we're at part 2, and I want to unify some concepts that I'm sure you're carrying around, but maybe haven't thought about together as much as we need for you to do. Okay, so it's going to be about chromosomes, maps, and a new technique, well, the technique's not new, new applications of a technique called FISH in order to learn more about chromosomes. Okay, so conceptually, I have this question, what do we mean by a chromosome? Okay, a chromosome might be a particular chromosome in a particular cell that you happen to be looking at in a karyotype. That thing is a chromosome for sure. But when we talk about chromosomes in the more general sense, we're talking about all examples of that chromosome that are out there in the population. So we can speak of that chromosome in terms of its genetic map or its sequence, realizing that that's pertinent to all examples, or at least most examples of that particular chromosome. Here's an example of a human X chromosome showing the G-banding patterns like we saw last time. Okay, it's hard to imagine that this chromosome actually contains about 155 million base pairs of DNA, and within all that DNA, there's only approximately 4,650 protein coding genes. Okay, importantly, this chromosome which everyone has two of, or they have one or two, depending if they're male or female, the genes are there in the same order and same relative locations compared to other examples of that same chromosome. It's important to realize that the genes are not shown on a G-banded pattern in this chromosome. That's a cytogenetic thing. It's not a genetic DNA thing, and we have to have other ways of representing and talking about the locations of the genes on this chromosome. Okay, so the concept of map is that each gene has a home on one of the chromosomes. And we can describe a chromosome or a genome in terms of its map, which is to say the locations of the various genes and where they're expected to be found. Genes have their normal locations on most examples of each of these chromosomes. Right. So the concept of chromosome, allele, and map are related to each other in that alleles are different versions of the same gene, and they occupy equivalent locations on different versions of the same chromosome. The map is where the genes are expected to be found, and the alleles are different versions of any gene at that location. Okay, so here's a good example that I like. This is the X chromosome again, and there are a lot of mutations that lead to disease, so everybody's interested in tracking out where these various disease genes are located and knowing what kinds of mutations can cause the disease. Okay, so the example I want to talk about a little is hemophilia A, which is caused by a deficiency in one particular gene on the X chromosome, and we can see it on this map where the location of that gene can be found. Hemophilia A is caused by mutations in the gene encoding factor VIII. Okay, this factor VIII protein is essential for blood coagulation. Okay, when we say that, we are saying the gene or DNA sequence that produces the mRNA, that produces the protein, is located on the X chromosome at the location shown on the map on the previous slide. Now, most people or most examples of the X chromosome contain active versions of this gene that produce the active useful protein that we need to have proper blood clotting. Now, some alleles of this gene encode inactive proteins, and those are the ones that lead to disease. Okay, it's the same gene that's abnormal that its deficiency results in disease. And it is allelic to the wild-type allele, which encodes the active protein, and that's what occurs on most copies of this X chromosome. Okay, here's a really scary picture of disease genes on the X chromosome. You can't read this, I can't read it either. But the idea is that, remember, there's 4,600 or so protein coding genes on the chromosome. There's lots of ways to cause disease, and these are just some of the diseases that are known to result from mutations that occur in genes on the X. So this list of diseases can be considered a genetic or genic map of this chromosome. And as you can see, they're pointing to various locations within the chromosome where the gene is typically found. So you can't look at a chromosome and list all 4,600 genes all at once. So instead, usually a few selected genes are chosen as markers or landmarks to be able to talk about particular sections of the chromosome. Okay, so here's a map where we've got selected genes that are shown on all of the 23 chromosomes, and they're just used as landmarks or markers for that chromosome. It's very representational. Okay, so now I'd like to talk a little bit about a technique called FISH, which is fluorescence in situ hybridization, and it has many uses, one of which is to locate genes on chromosomes. This is from the NIH. So the idea is that we have, of course, recombinant DNA. We've got plenty of ways to acquire and purify any particular piece of DNA from the human genome. And we're going to use that DNA to find its complement on a chromosome, right? So what we're going to do is get our DNA, which is referred to as our probe, label it with a fluorescent dye, which we're later going to be able to see, and then using our cells on our slide, denature and hybridize the chromosomes in such a way that the probe, which is labeled, can locate and bind to its complement on the chromosome, thereby enabling us to see the location on the chromosome where that DNA sequence is normally found. So our probe might be a purified sequence, just one, or it sometimes is a very complex mixture of sequences, depending on what you're trying to see. It's best if it's single-stranded, but double-stranded ones are used. But it's important to know that it can find its complement in an amazingly complex mixture, and that it's visualized because it's tagged with a compound which emits a color at a particular wavelength. Now, this is observed using very expensive fluorescence microscopes, but it enables us to see the locations of the complementary DNA on the chromosome. Okay, so the cells can be either in metaphase or interphase in this particular case. They must be permeabilized and fixed to the slide. And it's kind of amazing, given the number of proteins and histones and whatnot that are there on those chromosomes, that mild denaturation is sufficient to open up the DNA so that the strands can actually partially separate and the probe can actually bind to and remain bound to its complement on the slide. So it's a hybridization type thing, but it's on a slide rather than in solution. So then the excess probe is washed off and the cells are observed in the microscope. Here's a simple, easy to see example where we're going to detect the DNA sequence found at the extreme ends of chromosomes in the telomeres. Remember that telomeres are specialized DNA sequence and they're found at the ends of all the chromosomes. And in human, the telomere repeat sequence is written 5' TTA GGG 3'. This sequence is repeated thousands of times at the end of the chromosome, and we're going to be able to see these using FISH because we know the sequence that we're looking for and we know where it should be located. So knowing the sequence, we can create a single-stranded labeled probe, has the sequence written as shown, which is complementary to the TTA-GGG sequence that's present at the ends of all of our chromosomes. Okay, this probe is going to be labeled and shown in yellow on the slide so that we can later actually see in the microscope where the probe is bound and be able to see or observe the presence of telomeres on all of our chromosomes. Notice that the sequence here is written complementary to the telomere sequence, and I wrote it backwards so you could see where the complementarity actually occurred. Okay, so here's a cute picture. The telomeres are the little dots where the probe hybridized, and you can see there's two little dots at each end of each of these chromosomes. The DNA itself is stained with a different stain called DAPI, which is a generic DNA stain, which we can see by its blue fluorescence. So we can see the light blue chromosomes and the yellow dots, which will tell us the telomeres. Okay, a more sophisticated type of application is something called chromosome painting, which I think is absolutely astonishing. That what is used is complex probes or complex mixtures of probes that are specific to each chromosome. Remember, we do know what sequence comes from where now, so it's a simple matter to identify which sequences will match any particular chromosome and mix different mixtures, one for each of the chromosomes we're going to be trying to see. So these are all collectively going to be labeled the same color, and they're going to hybridize to detect their complements on the chromosomes. Now, if you want to detect different chromosomes, you simply use probes for that chromosome, but label them with a different fluorescent molecule that will be able to be distinguished from the color of the first one. So here's a cool one where we have 6 different chromosomes that were hybridized together in a spread by 6 pools of probes specific for each of those chromosomes and labeled in a different way with different fluorescent tags and observed in the fluorescent microscope. And so you can see a diploid, you've got 2 green ones, 2 blue ones, 2 red ones, 2 yellow ones, etc. Okay, and a very sophisticated and almost amazing, definitely amazing application of this is something called spectral karyotyping. So we saw the telomeres as little dots. We saw the 6 chromosomes in the previous slide. In this spectral karyotyping, this whole analysis is extended to include all of the 23 pairs of chromosomes and to set up probes and conditions so that each one can be distinguished from all the others. Okay, now it's not really 23 different colors of probes, unfortunately. It's different ratios and a very complicated image analysis setup is used to distinguish what combination of fluors is being seen in each. So it's not a simple, straightforward, yeah, there's 23 colors, because there's not. But it's very sophisticated software to analyze these images, and what we're seeing is called really pseudo-colored, but it's still astonishing, more astonishing. Okay, so here's a picture of a spread of human chromosomes that have been painted with a very complicated mixture of probes as shown, and those are arranged to form a typical karyotype simply based on the colors of the different chromosomes. It's easy to pick out the blue ones, it's easy to pick out the 2 5s because they're the same color of purple, and you can easily see the green X's, etc. So it's really an astonishing technique that enables us to do all this. Okay, in the picture, it's interesting, that's an interphase nucleus at the top. And that's, you can see the chromosomes occupying their own territories within the nucleus before they've condensed for mitosis. So that's what they would look like during interphase, as opposed to the mitotic spread that you're seeing here. Okay, here's a spectral karyotype of a lung cancer cell line, and you can see how outlandishly aneuploid and messed up these chromosomes really are. You can see the rearrangements very clearly by the colors and the different numbers of chromosomes. There's some trisomies and there's a bunch of translocations, and it's a clear mess. And it's obvious that this poor cell line has undergone many, many rearrangements to generate a karyotype that's as abnormal as this. This is not an extreme case, but tumor cells are very often aneuploid and generally have other visible chromosome rearrangements. Okay, so now we're going to look closely at that messed up karyotype and look for specific examples of translocations and trisomies. We need to look at various cases where there's 3 or 1, or cases where the 2 different colors seem to be attached to the same centromere as indication of translocation. So take a good look at the karyotype. And that's the end.